Pleasure to introduce Jasmine Canton. I think I got that right. Jasmine did her undergraduate degree in psychology and her master's at the University of South Alabama, and then she did her PhD with Brett, right? At Brett Clements. Okay, at the University of Georgia. And I've known Brett, her mentor, for a long time when he was back in San Diego. And then she did her postdoc work out in Minnesota with Calvin, who most of us know from our collaborations with the University of Minnesota. And now she's, of course, in Hawaii, or in Honolulu, at Neurobehavioral Research, Inc., which is a similar type of a place as this. It's a small, I think it's a nonprofit that George set up, right? Oh, it's for-profit? Okay, maybe they got a little better than us. Anyway, they're making money, apparently. And so a for-profit established to help do neuroscience, neurocognitive, kind of research. And George, is it Feinberg? Fines.
Yeah.
Fine. I always get it mixed up. Was on study section with me a while back, and then I talked about some of our data sets, etc. And then we decided that it might be fun to have some of his scientists come out, or you guys come out and talk to us about what you're doing, because we have a lot of overlap in our work in addictions and other things. So Jasmine's going to speak to us today about resting state, one of our favorite things, differences in abstinent alcoholics. And so they flew a long way from Hawaii, so let's give them a warm New Mexico welcome for traveling all the way.
So first, thank you for letting us share our research findings here. Everybody with Metro has been very welcoming, so thank you. So today, I'll talk about resting state differences in abstinent alcoholics. And the brain's functional organization is reflected in the ongoing synchronized firing of neighboring and also distant cluster of neurons. So the best way to measure ongoing synchronized firing so far is by measuring it during rest. And in our lab, we have termed this resting state synchrony, so RSS. So so what do the resting state networks do? What do they tell us? So they reflect interactions within cognitively relevant functional networks. And they can be thought of as a blueprint that constrains the responses to the external world. So for example, if somebody within an executive control network has strong synchrony during rest, it's very likely that when the person performs a task that uses the same network will perform better than people that don't have that same synchrony. So our main question is, how is resting state synchrony different in clinical populations? And right now we're interested in people that have alcohol dependence. So alcohol dependence is a disorder that is characterized by overwhelming appetitive drive and a lack of executive control. And I've heard from people talking here that alcohol dependence is also a big problem here as it is in Hawaii. So, for example, people that have alcohol dependence have a strong drive, an uncontrollable drive to ingest alcohol, and they lack the executive control to stop ingesting alcohol, even though there are negative consequences. So we were interested to see how these behavioral aspects, what are the neural networks that mediate these behavioral aspects. And there are neuroimaging studies that have identified brain activity corresponding to these behavioral aspects in alcoholics. So studies, mainly the fMRI studies that have used cue reactivity tasks, they have identified that alcoholics show increased activity in appetitive drive regions in the striatum, like nucleus accumbens, caudate, putamen. And they also show less activity in executive control regions when needed. For example, in dorsolateral prefrontal cortex. So most of these studies have been conducted in people that are actively drinking or in people that are recently abstinent. But we have noted in our lab that most of the people don't really get to be long-term abstinent. And that is something that we really want to study because it will be a model for people that are actively drinking or recently abstinent to be able to see how their brain functions, which it could be something that people would be useful for treatment, potentially for treatment, and like cognitive behavioral treatment, and I'll talk about that later. So only a small percentage of alcoholics reach successful abstinence. If somebody goes to treatment, usually it's about 25 or 30%, and it depends on what program they are. There's a program in Hawaii that they claim that they have about 70% successful rate, but it all depends on what program they're into. But usually it's a small percentage of people that actually get to be long-term abstinent. So that's our interest. What are the brain functional characteristics of long-term abstinence? Do they also show this exaggerated engagement of appetitive drive regions? And do they have a lack of engagement of the executive control regions? So in our lab, we have conducted 2 studies to investigate whether resting state synchrony in appetitive drive and executive control networks are different in abstinent alcoholics when compared to non-substance abusing controls. So to do this, we have 2 studies so far. The first study is where we compared 23 non-substance abusing controls to 23 long-term abstinent alcoholics. They were comparable in age and gender. And the long-term abstinent alcoholics, they needed to be more than 18 months abstinent. And the range was between 2 and 10 years of abstinence. So usually after this long time of abstinence, it's very rare that they relapse. And the long-term abstinent alcoholics, they also had no drug comorbidity. They used drugs, but recreationally, and they were not dependent on those. In study 2, we added another sample, which are the short-term abstinent alcoholics, who needed to be between 6 to 10 weeks of abstinence. So they were beyond the detoxification and the supposedly from the withdrawal symptoms, but they were still recently abstinent. And they also had no drug comorbidity. Another thing that the short-term and long-term abstinent, they didn't differentiate in how much they drank in average per month, how much they drank when they were peak drinking, the age when they started drinking heavily. The groups were not significantly different. So for the first study, we comparing the controls to the long-term abstinent alcoholics are based on reports of compensatory mechanisms in long-term abstinent alcoholics. We hypothesized that long-term abstinent alcoholics would show either a similar pattern of resting state synchrony as controls, or that they would show some kind of compensatory mechanism that would facilitate abstinence. So we expected that maybe they would show enhanced resting state synchrony of executive control regions and then reduced synchrony of appetitive drive regions. And then also another hypothesis was, given the relationship between resting state synchrony and behavior, we expected that the strength of the resting state synchrony in executive control regions would be correlated to performance in a task that requires executive control. And I'll talk about the task in a minute. So this is a lot of information, and I can get into it more in detail if required, but for the scans, people would go into the scanner with their eyes closed, and the scan lasted 4 minutes. And all this data was collected before we got to Hawaii. They have all this data for us to analyze. And we actually, it's a research dedicated scanner in Queens Medical Center, which is one of the biggest hospitals in Honolulu. And we collected the data, we processed it with slide standard correction, motion correction, spatial smoothing, intensity normalization, temporal band pass filtering, detrending, and then each individual data was denoised using ICA melodic. And then each individual data was registered to the T1 image and then to the MNI frame. And if anybody has questions along the way, I can answer too. So there are different ways that we can analyze resistant synchrony, and I have previously used independent component analysis and small role networks with Kelvin, and also region of interest analysis. And because in this case we had this predetermined hypothesis in which we expect that alcoholics would have differences in these specific networks, we decided to start with region of interest analysis. So here we focused on executive control and appetitive drive networks, and to generate those networks we chose seeds that are relevant to alcoholism. So here, the subgenual anterior cingulate seed is known to mediate executive control of emotion. And emotion is a very important aspect of behavior in alcoholism, which is very related to the ability to stay abstinent. So people that don't have the ability to regulate emotion are more likely to relapse. And then we also chose another seed in nucleus accumbens, which is a region, I think everybody's familiar with it, that is known to integrate the input from limbic and cortical regions, and it's known to process all reward information. So increased activity in nucleus accumbens have been found when people expect a reward, when they register a reward, or they give valuation to a reward. And also if people have motivation and learning to go search for a reward. So these are the two main seeds that we use to generate networks that have to do with executive control of emotion and also with reward processing. So after pre-processing the data, we did a multiple regression analysis where the average time series of each seed is correlated with all voxels in the brain, which generated a statistical map. And this map showed the positive or negative correlations with the seed. And then the values were transformed to standard Z scores. So then to look for differences between long-term abstinent alcoholics and controls, we did a t-test for each map that was generated for each seed. And then these resulting maps were thresholded and clustered using Monte Carlo simulations using AlphaSync for mapping. So we got very interesting results, and we wanted to share it, and any input on the results would be very valuable. So one thing was that we found that long-term abstinent alcoholics showed to have higher resting state synchrony of executive control regions, in this case, dorsolateral prefrontal cortex. So the green bars are the long-term abstinent alcoholics, and the blue bars are the controls. And they show to have significantly higher synchrony with both the subgenual anterior seed and the nucleus accumbens seed. So during rest, long-term abstinent alcoholics seem to keep engaged the synchrony of their executive control regions more than the controls, more than non-substance abusing controls. And then on the other hand, they showed lower resting state synchrony of appetitive drive regions. So people that are long-term abstinent alcoholics when compared to controls have attenuated synchrony of regions that are known to process reward. So again, the green bars are the long-term abstinent alcoholics and the blue bars are the controls. And they show significantly lower synchrony, in this case between thalamus and subgenual anterior cingulate, caudate and nucleus accumbens, and thalamus and nucleus accumbens. So caudate and nucleus accumbens are regions that are known to mediate reward processing. Usually when people start using alcohol or drugs, the reward processing is mainly in nucleus accumbens, and then when it becomes a habit, a compulsion, it moves into dorsal striatum, which is coded in putamen. And then thalamus is known to relay this information to the cortex to actually goal-directed behavior for it. So this was interesting that we found increased synchrony of executive control regions and then decreased synchrony of regions that are known to process reward during rest. So from here, we hypothesized that this higher synchrony of executive control regions combined with the lower synchrony of appetitive drive regions may represent some kind of compensatory mechanism that helps, that facilitates long-term abstinent alcoholics to remain abstinent. But we wanted to see how does this relate to behavior. So we have a task as part of the CANTA battery that's very similar to the Wisconsin card sort test, where people have to choose between two arbitrary options, and then they have to figure out the rule, but then at some point the rule changes. So then they have to figure out that the rule has changed and adapt their responses. And so this is a very important task for alcoholics because this is what they actually have to do in real life. They have learned to respond, to go for the reward, to go for the alcohol, but then because they want to be abstinent, they have to change their behavior and they have to be flexible and adapt their responses to the environment which they had already learned before. They have to adapt it so that they can stay abstinent. So we thought this was a good task to look at behavior. So this is the correlation that we found, that here the x-axis is performance in the IED task. So the higher the number, the better they perform, the less number of errors that they have. And then in the y-axis is the synchrony between nucleus accumbens and dorsolateral prefrontal cortex. So only the long-term abstinent alcoholics show this correlation, where people that have the stronger synchrony between nucleus accumbens and dorsolateral prefrontal cortex, they perform better in this task. And the one that had lower, they perform worse. So this kind of gives more confirming evidence saying that it's important to have that synchrony, that it may also mediate better performance in a task that requires executive function.
I'm sorry, can you tell me what the arrow is defined as? I don't remember the task.
Yeah, so in the task, there are distractors. So there are rules that they have to learn, and then there comes, within the rules, there are distractors that you have to figure out whether the rule, let me see how I can explain it better, whether the rule is because of that distractor, or if the rule is because of the underlying rule. So there are two different aspects that two different cues that you go to find about the rule. Is that so? Then there's another cue that will distract you to see whether you think that the rule is because of that cue. Is that clear enough? It's kind of so then whenever they figure out the rule, there are these other cues that keep changing. So whenever they respond according to that distractor, that's considered an error. Is that clear enough?
Yeah, so it's like maybe kind of that works, thank you.
Yeah, so that's the difference with the Wisconsin card sorters. It doesn't have those distractors. But then here, like, better performance means that they didn't pay attention to the distractors to come up with the rule. So after we found this in long-term abstinent alcoholics, we wanted to see whether these same differences can be found in short-term abstinent alcoholics. So again, just to point again, so these are 27 short-term abstinent alcoholics who have been abstinent between 6 to 10 weeks. They have no drug comorbidity and they were comparable in age and gender to the other two groups. So here we had three hypotheses. First, that we thought that if these changes, these differences in resting state synchrony have to do with how long they've been abstinent, we expected that short-term abstinent alcoholics might be intermediate between controls and long-term abstinence. And also we expected that short-term abstinent alcoholics might show similar correlations between strength of resting state synchrony executive control network and the task. And also because of the recency of the short-term abstinent alcoholics of having ingested alcohol so in a short term, we expected that they would still have craving and they would still have appetitive drive. So we hypothesized that short-term abstinent alcoholics would have a correlation between resting state synchrony and appetitive drive and behavior that ruled by appetitive drive. So all the individual analysis was the same as long-term abstinent and controls. But then because we wanted to focus our investigation whether the short-term abstinent alcoholics showed the same differences as long-term abstinent, we focused only on the clusters where the long-term abstinent alcoholics showed differences and extracted the mean z-scores from those clusters. So this is the same results that I showed you before, where the long-term abstinent alcoholics showed significant difference between with the controls, and then we extracted the z-scores from these same clusters and saw what's the strength of resting state synchrony in short-term abstinent alcoholics, which are the tan bars. So it was interesting that we found that it was intermediate. They have a very big variation because we assume that some of the short-term abstinent alcoholics are going to become long-term and some are going to relapse. So there's a big variability in this group. And so we found intermediate in this network, and we also found intermediate resting state synchrony in the appetitive drive networks. So we found that in the same clusters where the long-term abstinent alcoholics showed differences, the short-term abstinent alcoholics were intermediate, and they were not significantly different from the controls. So based on these results, we thought that the intermediate resting state synchrony of executive control regions and appetitive drive regions may indicate that there's a linear progression of the strength of resting state synchrony depending on the length of abstinence. But to be able to really claim this, we need longitudinal data, which were actually these same short-term abstinent alcoholics we had collected follow-up data 6 months after, and we're going to go into analyzing that next. So another question was, are these differences related to the mentioned behavior to the same task, the IED task, in short-term abstinent alcoholics? And we found a similar correlation where the short-term abstinent alcoholics, which each dot is one individual, they showed that the synchrony of executive control regions was correlated with performance in a task that requires executive control. So stronger synchrony was correlated with better performance in the task. So then we also collect data about psychiatric comorbidity in all of our subjects. And one aspect of one clinical aspect was that short-term abstinent alcoholics showed significantly higher current antisocial personality disorder symptom counts than long-term abstinent alcoholics. The long-term abstinent alcoholics did not differ from controls in the count of antisocial personality disorder, but the short-term abstinent alcoholics did, and they had significantly higher than the long-term abstinent. So we wanted to see whether this difference in current antisocial personality symptom count was correlated to resting state synchrony. And expected that because antisocial personality disorder is a lack of restraint on appetitive drive behavior, it would be related to appetitive drive region, so we did the correlation between so the x-axis is the higher the number, the more current symptom counts of ASPD. And then the y-axis is the higher the number, the stronger the synchrony within appetitive drive regions. So we found that people that have stronger synchrony of appetitive drive regions during rest, they have a higher count of antisocial personality disorder symptom count. So from these studies, study 1 and 2, we found that these may be adaptive differences that abstinent alcoholics have, where the strength of resistance synchrony may help them facilitate abstinence. So we found that resistance synchrony of executive control regions is higher in alcoholics with longer lengths of abstinence when compared to controls, and resistance synchrony of appetitive drive regions is lower than controls with abstinence, with longer abstinence. I think I said it backwards. But there it is. So the longer you're abstinent, more executive control and less appetitive drive region engagement. So this is our main conclusion, is that functional brain organization in abstinent alcoholics is different at different stages of abstinence. And to be able to say that there are actually changes, we would need longitudinal data. But this suggests that there's adaptive mechanism that may support behavioral changes to help maintain abstinence. So for future plans is that to look into the longitudinal data and see if this is true within subjects. And another thing is to confirm findings with other analysis methodologies like independent component analysis, and look at identify components that are known that, for example, frontal and parietal component or basal ganglia component. And also investigate whether this is specific, these findings are specific to alcoholics, or is it that people that have high appetitive drive, for example, psychopaths, they also show higher resting state synchrony between reward regions. And maybe that has nothing to do with alcohol or drugs, but from what I found from that, it seems like there's not that many psychopaths without alcohol and drugs. So that might be a little hard to disentangle, but we would still try to address that question, whether this is specific to alcoholic, to a substance abuse, or it's also other people that have problems with appetitive drive, if it happens too. And I wanted to thank the people that helped us collect data in Oahu, and our physicist in Queens, Andy Stenger, and our PI, our head PI, which is George Fine in our families.
So we can entertain questions?
Yeah.
So have you looked at all at the short-term abstinent alcoholics that relapsed since?
Actually, we actually we have a paper coming out in cerebral cortex where the short-term abstinent alcoholics I have here, they're alcohol only. They don't have any drug dependent comorbidity. But the other paper that's coming out, it's short-term abstinent alcoholics with alcohol and drug comorbidity. And it's 69 short-term abstinence. We looked at resting state at baseline, and then we followed up a year after, asked them whether they relapsed or not. And then there were 40 that remained abstinent and 29 that relapsed. And then we found differences in the resting state synchrony between the abstinent and relapsers. And we actually did a binary logistic regression where you can actually predict who's going to relapse or not. based on the resting state synchrony.
And that was that resting state synchrony involving the DLPFC or involving
It was actually like a more generalized resting state synchrony. All networks showed. We had executive control network and reward network. And we also did a network with insula and a visual network just to see if it's only these networks that they showed decreased synchrony, but it was an overall whole brain lower synchrony. That's what predicted who would relapse or not. It was for different networks, yeah.
When you examined the correlation between behavioral performance and synchrony, were you also showing us the control subject's performance there?
No.
How did they do?
They didn't show any correlation.
Okay.
And I think it's because they have like a ceiling, so they don't have that much variability on their performance. So they were, if they would be like more to one end, you know, all clustered in one end because they just perform a lot better. There's not much variability. So they didn't show any significant correlation.
So one thing I wonder is if you include all the subjects in the same correlation, whether the control subjects would in fact show more synchrony. I mean, we might expect that they have better ability to control their behavior. So if
to put everybody as just one whole group and see how, that's a good idea. Yeah, I'll look into that.
Related to his question, so if I remember correctly, your synchrony in this control network is lower in controls, higher than that. So it's basically look like the composition for the alcoholics will try to control and use more force or more activities to have better control rather than preferring similar controls. I think the question I have is, if I understand correctly, you choose two seed, and the one seed is try to capture the executive control network, and the other seed is try to capture the drive network.
Right.
But your DLPFC show connection in both seed to both seed.
Yeah, it showed. So the DLPFC was significantly higher synchrony with both seeds, with the nucleus accumbens and the here, and the subgenual anterior singular.
So if you understand that right, so you have DLPFC is highly connected to both like networks. So one thing is how do you explain that? But the other thing is, can you use DLPFC, look like there's more sensitive region, use it as a seed to say look at the connection with whole brain.
Right. Yeah. So one way that we interpreted these findings was that long-term abstinent alcoholics need to have executive control, higher executive control, which is dorsolateral prefrontal cortex, higher executive control with subgenual anterior cingulate, and also with nucleus accumbens. So it seems like in overall, people that are long-term abstinent alcoholics need to engage executive control with a lot of networks during rest. So that's like the theoretical. But then right now I'm doing that, like deciding whether to use dorsolateral prefrontal cortex as a seed. But the problem is that it's different regions, and it would be like I'm still deciding which region to use because there's pulmonary 9 and 10 and 46 and both sides. So so that's one thing that I was thinking about doing to use dorsolateral prefrontal cortex. That and insula, I thought that would be another good seed to use because increasingly people are identifying insula as a region that has to do with craving and interoceptive awareness. So those two seeds I've been thinking about using too. But then, there's so many ways to analyze this.
Sorry, another question is for the short term. Last result you presented the increase of the symptomy in short term alcoholics. Their performance is positively related to increase of the synchrony there, but also same time possibly related to antisocial personalities or common there.
Yeah, but this is, so it was positively correlated, the synchrony of dorsolateral prefrontal cortex here, but here is positively correlated with the synchrony of nucleus accumbens. So here, the strength of synchrony here is of appetitive divergence between nucleus accumbens and caudate, and the other one is with executive control regions between subgenial anterior cingulate and dorsolateral prefrontal. No, nucleus accumbens and dorsolateral prefrontal cortex. Yeah. So yeah, it's the same direction, but it's two different resistive synchronies.
And how do you, because you showed that in the long term, since alcoholics, their antisocial is like normal case,
Right.
And then, but they're also, do they also show higher sympathy of this resistance?
No, they don't. They didn't show that. Yeah. And that's another question that we have, that our long-term abstinent alcoholics, we don't know if they have been self-selected, if it's a group that because they have stronger synchrony of dorsolateral prefrontal cortex, they were able to reach long-term abstinence, and we just have this selected group where the people that don't have that increased synchrony, they actually never reach long-term abstinence, and so we don't have, you know. So it's, we don't know if these differences are because of self-selected, and that's why they actually got to be long-term abstinent alcoholics, or if it's, this is a result of being abstinent. We can't answer that from this. Any other questions?
Along those lines, getting back to the a little bit too, it would be interesting in the short-term alcoholics if you saw that the ones who had greater activity of the DLPFC and anterior cingulate were those who ended up successful in abstinence.
Yeah. So we do have follow-up data, because the one that I told you about, it was just looking at the baseline resistive synchrony, but we have follow-up data on the short, on those same, and we have to see whether there's what's the change in resistive synchrony from time 1 to time 2. But yeah, we're having problems, like server problems, so I haven't gotten to look at that data yet. Yeah.
You may have said this already, but were these people in treatment?
They're all into outpatient groups.
Okay.
So the Alcohol Anonymous and other groups, but they're not, the long-term abstinent, they're not in treatment, but they still meet. And that's how they recruit them, because they, the rest of their lives, they will go to some kind of support group. So they still meet, and that's where they actually put the ads, and that's where we get them. Because, but yeah, they're none of them are in under treatment. They're just being counseled and have their mentors to help them along, but they're not in any treatment facility.
All right, well, thanks again, Jasmine.
Thank you.